how much mutational burden is very important in a cell's life on a cancer pathway. The more is the mutation load, the chances are the cancer is going to get nastier, and it may not be sufficient for a single drug to function to restrict its growth. So I hope that is clear. Here is the summary for this lecture. And before I move on, I would want to go to the questions. If there are any questions that you want to ask on what we discuss in terms of the tumor suppressor genes or the other gene, the oncogenes. If there are any questions, please feel free to ask me now. Otherwise, we can simply switch to a discussion on this tutorial on omics, which I'll try to keep short, of course, because a lot of it has been very elegantly discussed by my colleague, Dr. Aziz. Well, if there aren't any questions, please feel free to. Okay, there is a question now. Padmavati is asking, and please feel free to ask me any career-related questions. She finished her PhD in cancer research in IIT. I found your research. Thank you very much for your kind words, Padmavati. Postdoctoral opportunities. So at the moment, I don't have any availability for a postdoc in my lab due to the funding crunch. So I don't have the postdoc I had in the lab. Recruiting a postdoc is actually quite difficult and different in this country because they need to bring budget to cover the indirect cost, meaning the electricity, the heating. It's a cold country. In India, most labs don't really consider the government. In fact, does not give any indirect cost. But in countries like UK, US, you need to have substantial indirect costs. So at the moment, I don't have it, but I can tell you something. If you go to this website, you have the right profile. You will get a response positive, I hope. But also, if it is not successful, for any job in UK, you will get an answer whether your application was successful or not, which is quite good because once you apply, there are many countries I have heard where people don't even respond to emails. So at least if you're applying in UK, I can tell you, you will get a response after the due date has passed. A shortlist has been formed. Whether you're shortlisted, well and good. If you're not shortlisted, they will send you an apology letter saying that they're sorry, your application has not been successful. So there is a very structured interview process as well here. They will do first interview, which will be shorter. Then they will do a second interview, which sometimes will be on site. So they might invite you to come here and do an interview, meet the team, spend the whole day, see whether this is something you really want to spend your 3 years of your life doing, because it's like a make or break situation. Those important first years of postdocs are very important in setting the scene for you to become a successful academic faculty or a scientist in industry, for example. So yes, please do apply on most of the jobs in the UK are published on this website. Are there any questions, or shall we start? No problem. Okay. I can start the omics session today, and I promise I'll keep it brief because I know you have an overdose of omics by myself, and more importantly, very excellent talk by Dr. Aziz. So let's start this session. I want you to ask me questions as you feel fit by typing them in the box. So, this is a regular central dogma of molecular biology. DNA makes RNA as a result of transcription. RNA, not all RNA become protein. As we know, there are lots of RNA. In fact, majority of them, different types, which never make protein. Only the mRNA make protein. You know that, right? And many of the proteins, we know most of the enzymes are nothing but proteins, and they are involved in cellular metabolic pathways, and they can lead to formation of new metabolites as a result. So sometimes through metabolic pathways, you can get hints of which proteins are active in a cell. So studying them, looking at P53, looking at ATM, I was telling you ATM phosphorylates P53, well, good. But I also told you that ATM also phosphorylates 800 odd other proteins in a cell. So there are a number of substrates of ATM kinase in a cell. P53 is just one of them. To make all of this sense altogether at a system level requires lots of skills like RNA sequencing, mass spectrometry, which I believe Dr. Aziz discussed with you previously today. So transcriptomics is nothing but to measure the level of the mRNA of different genes at once in a cell. That is what we study when we study transcriptomics. The way to study is through either microarray, which are still in use but have been phased out mostly and replaced by a technique called RNA sequencing. Then you have proteomics, which is something my lab currently focuses a lot on studying proteomics to measure the abundance of proteins, because ultimately proteins are the ones which are the drivers of cancer, and more so because proteins are the ones which are comparatively easier to target compared to RNA or DNA. Of course, there are lots of non-coding RNAs, microRNAs that people are trying to target, but the one which has been most successful in targeted therapy is to target proteins, and the proteins that cause cancer in uh, industry and hospitals, of course, clinical systems. Metabolomics is nothing but to study the metabolite, what metabolites are being formed by a cell, what types are they. So ohm is a simple word that you can use to define an entire set of proteins. So gene, you can say it's a genome. Protein, you just said proteome, it becomes the entire set. So that is what is defined by ohm, and the study of it is called omics. I'll keep it short. So you can apply omics to define cellular pathway these days. For example, have a look here. This is a TCA cycle or Krebs cycle as we know it. There are different enzymes of the cycle which will become active when the substrates are present. So you can, with the help of the transcriptomics, proteomics technology, you can work out all the mRNAs which are overexpressed in cells in a specific stimulus conditions. These are the enzymes which the cell is making. These are the proteins, and these are the metabolites which are being formed. So what has happened with transcriptome, proteome, and such a discovery of these technologies is to become much easier to do discovery research. But I think we are getting more intellectually weakened, especially with the advent of artificial intelligence and the reliance. There are reports that we are not learning as fast as we did before, thanks to the AI. So I would say still human intelligence, there is no substitute to human intelligence. Um, and You can see here in terms of this, to discover scientists, Hans Adolf Grabe, to describe this biochemical pathway, to see which enzymes are present, what are their cofactors when they are incubated with the substrate, several of them, what products are formed, must have taken not only their lifetimes, but also required a lot of intelligence. Now there are things which you can do using omics is to identify all of them in one go. I'll show you in an example. But within this Krebs cycle, you can see there is this enzyme called IDH. Mutations in IDH are very common. If this has not been discussed today, then I will certainly catch up on this tomorrow. You can see isocitrate dehydrogenase. The gene makes a transcript, then a protein is made, the isocitrate dehydrogenase protein, which can convert this isocitrate into a product called alpha ketoglutarate. So you can see that when isocitrate dehydrogenase gene becomes active, you would see that the cell will have higher levels of alpha ketoglutarate compared to isocitrate. You block this pathway, you will see that the isocitrate will start to accumulate in a cell. So these biochemical reactions are painstakingly difficult to carry out. Understand, make sense of them, interpret the data requires much higher level of intelligence to decipher these pathways compared to now we have tools at our disposal like these omics technologies which does uh, do facilitate the discovery research. But I have to say again, there is no equivalence of human intelligence, why these pathways are active, what happens, and there are a lot of things to do with these pathways, simple biochemical pathways in cancer research, which I can show you tomorrow. So you can see here this pathway as I gave you example of, and have a look here what happens. Why is it important that we study genomics, transcriptomics? One of the things you can see on the slide is part of small genome shown here. It contains two genes, gene A and B. So what you would expect normally is that you can gene A is more active in a cell. And when I say the is more active because it is making more mRNA. And what you can think that if there is more mRNA, we do qPCR a lot of time thinking that actually if you have more mRNA, you will have more protein. Meaning these are linear events. That's what we think, that more RNA simply means more protein. Actually, it is not true for majority of the human genes. That not a lot of RNA which you can detect using qPCR technique would translate into a lot of protein. Sometimes it is actually reverse, and I can give you some examples if you would like. And for example, gene B, it is not very active. When I say it's not very active, you must have seen from Dr. Hifzur Siddiqui's lecture today that genes which are not transcribed very well, they have epigenetic repression, they might make very little product in terms of mRNA, the transcript, and then it translates into very little, if at all, protein. But sometimes RNA is stabilized by different mechanisms in a cell, meaning despite having less RNA, you can have a lot of protein made by cells. And that's why I'm saying transcription and translation are not linear events in eukaryotes. In fact, even in prokaryotes, the same thing exists. But sometimes you may have a lot of RNA and it may not be translated into a viable protein abundant product. And that's why it is very important that we study the gene activation, what is called epigenomics, what we discuss, I think my colleague must have discussed today. And then we work out the levels, the abundance of transcripts of all genes using transcriptomics, and eventually what proteins and in what level they are being made by a cell. That is very important. So how do we establish this? I think my colleague must have discussed these, Dr. Aziz must have discussed this today in transcriptomics. So I will keep it short. Dr. Aziz, did you discuss these? Microarray and so on?
Yes, I did. I covered it in the
You have.
morning.
Okay.
And
So I will
qPCR,
skip.
I can give that a heads up as well tomorrow. But
Okay.
qPCR, I haven't
Okay.
told them in detail.
Okay, so I will skip this then. And you can see here, I will come to this example here. Uh, have a look what is happening here. You have, is that what you covered as well, Dr. Aziz? Sorry, microarray.
Yeah, I had a very similar
Ye-
slide.
Okay,
So
I
it's
will
covered
skip
the
this
color.
then.
Mm.
I will skip that then. Uh,
Yeah, you can
and
go
I'll
ahead.
go
This will
straight.
be a revision for them as well. So I think it's okay. Probably they
Okay,
didn't get it in the morning, so they can get it now.
okay, I can tell them one story about transcripts of these six transcripts shown on your screen. So lots of proteins change in a cell. When you take any drug, you take out, let alone drugs, you take out cells from incubator. and do a simple transcriptomic analysis, you'll find a lot of genes have actually changed in their expression. So it is a very dynamic process, and that's why what is happening in a body is sometimes, not sometimes, all the time, very difficult and different from what is happening in an incubator. And that's why we cannot really recapitulate what is happening to cells in terms of expressing specific transcripts. Uh, is this the same? Is this being honestly recapitulated in laboratory conditions? The answer is not really. And that's why many times we fail. As many as 95, 99% of the molecules would fail in phase 1 clinical trials, because there was not very strong science in discovery science, first of all, and many times because these cells don't cry when you give them drugs. Of course, you can look at some markers, but humans, if they experience any side effects of these drugs, one of the most common one is neutropenia, so decrease in neutrophil, which cell type? Neutropenia, I can't remember on top of my head. So specific decrease in lymphocytes, I think neutrophils. That happens in many cancers. You're not going to stop any clinical trial for that. But if a clinical trial leads to a death, which is defined, on a drug caused by a death, not in an accident, going in a car, then this would be a serious challenge for a clinical trialist to continue with the trial that will kill a clinical trial. So in terms of gene expression, it is not linear, and genes actually, as Dr. Aziz was saying, they're part of networks. They are regulated, and they regulate sometimes as a pathway. There are certain genes which sit upstream of these pathways. I was showing you examples of androgen receptor, which was regulating some genes downstream by binding to DNA. I'll discuss this tomorrow. Have a look at this example. Somebody generated a knockout of gene C, and now they, from a cell line, you can imagine this is a cancer cell line, and that is, they did the transcriptome profiling, and that is what they get. So when they look at these 6 genes, A, B, C, D, E, F, that's what they found. They were part of the, they don't know what sort of regulation they have, but have a look here in green bars. This is how the transcript for gene A, B, C, D, E, and F look like. Now, in a bar from a different cell line in which the gene C has been knocked out through genome engineering, genome editing, see what happens. These cells do not make any mRNA for gene C, meaning your experiment was successful. And you don't see any, the gene is gone, of course, the mRNA is gone, you don't make any mRNA. And you're seeing that this removal of gene C has no effect on gene A. It enhances the expression of gene B. It decreases the expression of gene D, E, and F. Can you start interpreting this data? What might be the role of gene C on these genes? Why do they look that they're changing the expression? You can instantly start to develop this idea as a biologist. I'm not talking as a computational biologist. I'm talking as a biologist. If I look at it, I would think, okay, it looks like this gene may not be relevant at all to this pathway, because if I block, if I get rid of C, there are no changes. I'm still making 100 copies of mRNA for gene A, sorry. And have a look here. Gene B has gone in expression much higher. It is making more copies in situations where the gene C has been deleted. Instantly, I should get an idea that gene C is actually causing a blockade of the expression of gene B somehow. So gene C appears, the product of gene C appears to be a repressor for gene B. Now, it may also be that this gene D, E, and F, 3 genes, they're decreased in expression when you get rid of gene C as a result of knockout. And you can see here the effect. All 3 genes go down in expression to some degree, 20-fold, 10-fold, 50-fold. And that gives you, again, something to think about. these genes may have been more reliant. For example, this must have been more reliant on the expression of gene C. Gene E was much more reliant than gene F. You only see 5%, sorry, 5 times drop in the expression from intact wild type cells to gene C knockout cells. There is a 5 times reduction in the expression of this transcript. Compare this with this, where there is a 15 times decrease in the expression of gene E when gene C is knocked out. So you can actually check the relative dependency of these genes on gene C here. So if I show you this pathway, you can actually start making this pathway. This is for your reference, the same as you saw on the previous slide. Hmm. Have a look what is happening. These are 6 genes. And remember, gene A was totally unaltered. We think this may not have been part of the pathway. Uh, have a look what is happening here. It turns out after biochemical experiments looking at interaction between the proteins, between the mRNA, that there are some interactions. What kind of interactions? Gene A drives the expression of gene C. Gene C drives the expression of these 3 genes, and gene E blocks the expression of gene B. So what is happening here is, if you get rid of this gene C, which we did in this knockout experiment, you can see gene C is totally gone. The mRNA is totally gone. When gene C is knocked out, you can see straight away gene D goes down in expression, so does gene F, and so does gene E. The effect, of course, was more pronounced on gene E, as you can see here, compared to gene D and gene F. This could have been one of many reasons, actually. But what is happening here, gene B now goes in expression. Why? Because gene E was suppressing the expression of gene B. By getting rid of gene C, what you've done is you have blocked the expression of gene E product, and that's why the gene B product now goes up, the mRNA, I mean. But you can see, I was telling you, based on this, that it looks like gene A is not really doing anything here, because gene A was totally the protein, sorry, the transcript, the mRNA was totally unaltered when gene C was present or absent. But actually, it looks like that gene A does regulate gene C because it sits upstream of gene C. So it is the major transcription factor, perhaps, which is regulating the expression. When you get rid of gene C, you can see here this upstream factor remains unaltered. So this is how you start to develop understanding of pathways. This is not something which you want to do with 20,000 transcripts of a cell, would you? And that's why we need smart, efficient computational biologists who can think out of the box to make sense of the data we get from a sequencer. Exactly. And they cannot really work alone. They need support from wet lab scientists who can tell them, start to make sense of this data, prove and validate the experiments that show that what I'm showing in a pathway are actually facts, in which a pathway is regulated, being upregulated or downregulated by some components of a similar pathway. So you can certainly apply this knowledge on transcriptome and develop expression profiles. I can give you this example here. Have a look what is happening. There are the 6 genes. Each one is represented here by this line, and you can see here, this is the on y-axis. Sorry, there is no axis shown as such, but you can see this is telling you the expression level of these 6 genes. When you treat the cells with glucose and take out the mRNA, do the qPCR analysis, you find that when the cells are treated with glucose, all of them go up in expression. You might think that this might be actually glycolysis genes. They may be the ones regulating the glycolysis. But is this how the glycolysis pathway was discovered? The answer is no. It required efficient, smart, excellent biochemists doing painstaking research, bit by bit, each enzyme being expressed, not expressed, purified from liver extract, tissue extracts, and using those enzymes, incubating them with different substrate, not knowing what could be the substrate, and then looking at the products, what is formed. quantifying them, and then uh, connecting these dots to create the pathway as we know glycolysis. But nowadays you can actually do this at astronomical scales. Have a look. This is an oversimplified example: glucose enhancing the expression. You use a different cell culture plate and treat this time with much higher levels of glucose, and you find similar things, that the expression of these genes is going up. Certainly you will get an idea that these genes may be regulating similar pathway. So this is a metabolic pathway, but something similar actually happens in cancer cells. You might find that there are genes which follow similar expression profiles, which have similar expression patterns in cancer. And what we do as computational biologists, I'm not one of them, my colleagues are, who I collaborate with, that we develop these expression profiles. So what you're seeing on your screen is a map. Basically, it is heat map. And what you are seeing in this heat map, each there are three individuals, so each column represents the expression of a particular gene in each individual, versus in healthy males versus patients of prostate cancer. So now you might ask me this question, perhaps many of you already know, who are advanced students in the academic ladder, is why would you get prostate tissue from healthy males? Well, whenever surgeons are doing surgeries, there is a principle of surgery. You don't leave anything behind. So you need to make sure that all the cancer has been taken out, has been excised, even at the cost of removing some nearby non-cancer part where you suspect there may be some cancer cells which are trying to invade through the organ. So the surgeons, if there is any surgeon on the call, they can elaborate in more detail. But the principle is you don't leave any cancer cell behind, and you need to make sure that the margins of the disease, there is no positive margin left when you cut the slice the tumor out from an organ. So that's why you will get this tissue. They will go to a pathologist and they will say, hang on a minute, yes, there is a tumor core. Most of these cells, they're all cancer. But when you start to cut the sides, the periphery of the tumors, you might realize that actually the margins are now clear. The tissue that you're getting is entirely normal tissue, meaning you have made sure that you've taken out all the tissue which is all clear now. And that actually is not thrown away, but actually you can isolate mRNA, do the RNA sequencing, and use those tissue either as benign tissue, or some people, in case of prostate cancer, will give out their tissue out of choice, because prostate gland, as we age, grows in size, and where it sits is underneath the bladder, as I mentioned, and it surrounds the urethra, meaning it is going to block the bladder being completely emptied. And as a result, these patients will go back to the toilet again, and they will still feel that their bladder is full. Those patients will undergo a procedure called radical prostatectomy. And that means their prostate gland will be completely removed. Those patients will have their studies, if they're part of any study, or if they give consent, then their RNA sequencing data can be, transcriptomic data can be obtained, and then compare with patients who have prostate cancer. Now, I want you to have a look here what is happening with the color. So the red color shows high expression of genes which are listed in these rows. This is one gene, gene A, for example, gene B, gene C, and so on. And the same expression in cancer conditions. So have a look here what is happening. These genes, set of genes, they are overexpressed in normal healthy males, but their expression has gone down in terms of mRNA expression has gone down in cancer. What do you think, what we covered in the last 2 hours, where would you categorize them? Are they oncogenes, or are they likely to be tumor suppressors? Their expression has gone down in prostate cancer patients. I would think that they're likely tumor suppressors, or genes which were keeping the organism safe from developing cancer. Have a look at the second subset here. What is happening? The expression of these genes, which are down regulated, as you can see, the color green means down regulation, low expression. They are actually overexpressed in cancer patients. Three of them are shown in this example, and that can actually form a signature. So these days, cancer is treated, that we have new therapies of cancer. Sometimes what people are looking at is at the specific signatures in cancer. What type of signatures are there? They can call them signature 1, 2, or 3, and based on this, patients can be put into specific clinical trials or a specific drug regimen based on the expression of particular phenotype. Imagine someone who does not have any HER2 overexpression being given a drug Herceptin. Would it be effective? No, it might have side effects. So this is certainly very important, and we can make these heat maps. This is one of the oversimplified heat maps. If you go and look at some of the literature on cancer genomics, clinical cancer genomics data, you will find that these are enormous maps can be generated for hundreds of patients from one or multiple trial centers, and genes can be mapped, and such plots can be formed, and from this you can deduce, you can get the signatures out of genes which are possibly the ones associated with cancer. So these are the ones, if you take them out and see what these genes might be, you might find that actually these are the ones, some of which are already known to cause cancer, some of which are already known to be oncogenes. So if I give you this example with the regards to the molecular subtyping, you can see here uh, there is something important which is called molecular subtyping. What you're seeing is called the IHC based data. This is how the patients, women who have breast cancer, are treated. Their cancer by pathologists in the lab, and this happens in every country, including India. Their cancer can be defined as having expression of estrogen receptor, progesterone receptor, or her receptor. This will be, if they have all three of them, we will call them triple positive. We don't really call them that, but we call them triple negative. Why? Because if you are triple negative, as we discussed this morning, morning UK time, triple negative actually have very poor prognosis, meaning the outcome of the therapy treatment is really, really bad. I showed you those graphs, Kaplan-Meier plots, and triple negative patients who have none of these actually do very badly. These tumors, pathologists can tell you these cells look like basal-like. They have specific cytokeratin markers which will allow them to categorize them as being basal-like. If you're luminal-like, then the chances are that you will respond very well to the therapy. And have a look, something else is happening here. If I'm giving you these two examples, basal and luminal-like, have a look at this basal-like says Ki67 negative. Now, those of you who don't know Ki67, it is a marker for proliferation. Any lab studying cancer, any clinic, any hospital doing biopsies, they will look at the proliferative index, how fast the tumor is growing. You have more Ki67, of course, the chances are that the tumor is fast proliferating. Many times it is a bad news, but have a look here. In this case here, where the prognosis, where the outcome of the disease is super bad, estrogen, progesterone, HER2, all three are missing, but Ki67 is also negative. They're not growing very fast. So does that mean that if you don't grow fast, you're not cancer? Well, it is cancer. It is not growing fast for now. It has probably gone into a state we call as senescent. In P53, I was showing you a slide. Those cells are still alive. They are just waiting out for the right time when the drug will not be put on them. There will be no systemic or other treatments so that they can actually get revived again. Have a look here. These cells don't divide. Many of the cancer therapies, especially chemotherapy, will depend on the ability of cancer cells to divide at a faster rate. You divide faster, you die faster as well. But in this case, these cells are not very much in a dividing phase, meaning any chemotherapy you throw at them is likely to be unsuccessful. So why am I telling you this in this omics is because at the moment, what the signatures we have, they work. We have only of these few handful of subtypes, 1, 2, 3, 4, 5 shown on this screen. And you can roughly divide them into good prognosis women who will do well and probably will have become cancer free, versus those who are likely to do bad. Sorry. So what you can appreciate is that with omics we may be able to make more precise subtypes and identify these signatures. In this case this is shown for prostate, but there are plenty of data. Have a look at this data for breast cancer, which is nothing but came out from omics of more than 1000 women. It's called metabrick data from Cambridge University, and that has a lot of data. I've typed it in the chat box, metabrick data for the clinical genomics of women with breast cancer. They have exome sequencing data, they have transcriptomics data as well. So coming to the last slide before I start showing you that platform, let me know if something is not clear. I can certainly uh, show you again those slides, but a lot of what we are going to discuss is going to be based and relied on your understanding of this last slide, which is here. Have a look what is happening here. This is super cool, looks excellent, and this is exactly what you're seeing is much more complex data, but similar data what I was showing you in this chart here. So what you are seeing here is the expression of androgen receptor. Why androgen receptor? Well, this is what I study for past almost more than 2 decades now. So you have patient samples, the RNA isolated, the RNA sequencing done. So what you can do is you can isolate mRNA and you can do RNA sequencing, and you can see there are more than 500 patients. You can see the scale. This is 50 samples. And what you can do as a computational biologist, you can make a spectrum of this where you can put these patients who have either very high expression of androgen receptor and patients who have very low levels of androgen receptor. Now you will tell me, hang on a minute, you were saying androgen receptor drives prostate cancer. Yes, it does. But like breast cancer, where I told you estrogen receptor drives breast cancer, HER2 drive cancer. We also have breast cancer phenotypes like TNBC, triple negative breast cancer, which we don't know what is the major driver of TNBC. And there is an intense research investigation to identify the drivers of TNBC. Likewise, here, there are prostate cancers which do not have any androgen receptor. And you can see within this data, these are about 70 patients who are negative or they have very low transcript levels of androgen receptor. Now, have a look at the second bar. So this allows you totally free without having a login, this Xena clinical genomics data set to allow you to study your own data as well as study the data which has been submitted as a part of this consortium, TCGA consortium. You can see the level of number of samples over 500 only for prostate. Your favorite cancer, have a look there. You will find it in that data set. And we will, in the second half, I'm going to stop soon. We will, in the second half of this tutorial, we will have a look at this platform. Have a look. This is a second gene called ARID1A. This is just not a random gene, but we know ARID1A is one of those genes which bind to androgen receptor protein. So protein binding with protein and regulating, promoting the function of androgen receptor. I should tell you a lot of these slides, and I did mention that what I was showing you was oversimplification. Many times proteins work in coordination. They bind to each other to drive specific activities. We saw with cyclin D, there were 3 proteins binding to it, but chances are that there are other hundreds of proteins binding to that complex, which we don't know yet, or somebody might have worked out. Androgen receptor, we have recently found binds to about 1100, so 1100 proteins are now known. We have established that bind to androgen receptor. Some of them were already known through biochemical experiments, through yeast-2 hybrids, mammalian hybrid systems used in 1990s. But now we know that actually there are lots of proteins which we never knew can bind to other proteins, especially the drivers. So in this case, ARID1A, you can see here, patients who have very high levels of androgen receptors, it seems they have very, very high levels of ARID1A. Conversely, patients who are very, very low in androgen receptor, they don't need ARID1A because ARID1A is needed to drive the activity of the androgen receptor. So they're very, very low in androgen receptor, they're very, very low in ARID1A. Makes perfect sense. Have a look at this third gene, A1BG. And A1BG is not something that we study in prostate cancer, but having high levels of A1BG is a good prognostic marker in liver cancer. Meaning, if you have lots of expression shown in red, those patients in liver cancer will do better. They will live longer compared to those patients who have low levels of A1BG. So you can think that A1BG may be a tumor suppressor. Who knows? So you can see here that what looks like here that there is an opposite correlation. Patients who have very low levels of androgen receptor have very high levels of A1BG, meaning it is possible that A1BG is a tumor suppressor even in this context. And androgen receptor was inhibiting the expression of A1BG. In tumors who don't have the androgen receptor, A1BG cannot be expressed. So you can see how complex these data sets are, and they can be used, patient data, to make real world discoveries thanks to platforms like Xena, clinical genomics platform, which I will discuss with you in about 10-15 minutes from now, and show you what we can do with the help of these platforms, clinical genomics studies, to study the transcript expression and see how the transcript for gene A, for example, correlate with gene B and gene C, and so on. So I would summarize this. I'm not going to read this out, but basically this is in a nutshell what we have covered so far in this omics tutorial. And with this, I will stop sharing my screen and go to the Xena platform after 10-15 minutes, unless you have any questions. Shall I stop sharing my screen? Dr. Aziz,
Yeah,
shall we
I
take it?
think your lecture was very complimentary to mine because you went in depth
That's great.
and I skimmed the surface. Uh,
That's
I
very
think
good. That
a nice
compliments
commentary.
very well. Thank you.
Although I told them that microarrays are obsolete, because I did that myself and then I no longer use it. But yeah,
No,
keeping
that's
you
true. Microarrays are not used. Uh, they're used for a specific purposes. For example, focus custom arrays for proteins. Immune arrays, more like they are not used these days. You're absolutely right. We use mostly RNA sequencing these days.
yes. So
So shall we reconvene in 13 minutes from now
okay.
and start? And or if you want, we can crack on and finish early because I realize I'm only starting my day and it's the end of the day for most of you. So if you want, I can continue or I can stop.
Well,
Your
that's
choice.
totally up to you. I think
I'm fine with anything.
okay. Then I think let's continue because
Okay.
taking break might drop some
Yeah.
of the candidates here. So
Okay. That's fine. I will stop sharing my screen, this presentation, and then I'll start presenting some of the platforms I'll show you today. Okay. So let me just start this. So first thing first, how would you know whether there are genes which are important for growth or not? Um, is there anyone who can think? Uh, is there any platform? So I will go to Xena as well. But does anyone know any platforms that they can use? to find out, okay, this is the cancer I'm talking about. This is gene X. This is part of my PhD project. Uh, Before I start a knockout experiment, how would you know that this is actually the driver or potential driver, possibly? And there are lots of information available on the web these days, supported by some excellent institutions around the world, where you can get such information. And let me show you my screen. How would you find that data? Dr. Aziz, did you cover that in your lecture?
You're talking about UCSC Jena, right?
No, can you see my screen?
Yes, cancer dependency map. Okay. I thought you were
Yeah,
talking about
did you show it to them?
no, no depth map miner, no cell model passport, no. So this all would
Okay,
be new for them.
great. I think we can access this here, or let me check. It would be on
You
Sanger.
put the web link for this
Sanger.
depth map.
That
Okay, so the cell lines.
that
So
map. Yeah, I think I'll go to.
you can type
This
the
is
web
really
address.
really cool. What do you can? Uh, Dr. Aziz, I think so. This is not something you've discussed. I think when it was started, you did attend the conference. I, you were once in Cambridge.
Yes, yes, yes. Hingston.
Yeah, I think so. The depth map is Hingston, yeah, it's a village near
Yeah, we use
Cambridge.
this depth map portal
Mm,
quite often. In
don't
fact,
think
one
we're
of my
bad about
PhD students
villages.
who has
That's what
worked
we
on
say.
depth map.
Oh, you do use.
Yes.
Oh, you do use it. Okay. So
Yeah.
depth map is an excellent platform to see what happens to the expression of your own genes. For example, if you have a project, you're working on it, and
Can you please,
as part of
sorry,
PhD, a
sorry
master's
to interrupt.
project,
Can you just type the address, web
please.
address on the message box so that if users want to
Okay,
follow?
let me do that. Yeah. So, so before you start spending time doing research, i- for example, making those hairpins, devising a knockout protocol or a siRNA protocol, and start inhibiting this protein in a cell based on some data that shows that this protein is overexpressed, you might as well actually look into this platform and see what is the impact on cancer cells of getting rid of removing through CRISPR this particular gene. I've provided the link. I will type in the name of my favorite gene unless somebody else has anything else to tell me. Uh, Androgen receptor, not the agonist. Let me type in AR. So androgen receptor, if you type in the symbol, it comes up. Now have a look here what is happening in these data set. First and foremost, you can see the location. I was telling you androgen receptor gene is located on chromosome X. These are some of the names, alternative symbols for the androgen receptor. What I want you, there are lots of information here, expression data, copy number data, mutational data, dependency data, structure, inhibitors. So you can actually pretty much uh, write a BSc thesis, at least if you study this more extensively. What I want you to do is see here, this is a perturbation effect. Perturbation simply in this term means that you're going to inhibit androgen receptor uh, gene expression and measuring the effect on cell growth. Have a look what happens here. So if I click here, that's what you're going to find. So this is 0, and then you're seeing there are different cancer types here. And even within different cancer types, if you go to prostate, you will find here that this is the cutoff here. And you can see in these two cell types, LNCAP 22, these are cell models. You know, there are different models of different cancer, which you can grow in an incubator in a lab. And you can see here, by getting rid of androgen receptor in this 22RNE1 cells, and by getting rid of androgen receptor in LNCAP cells, you can actually diminish their growth. So these are the two cells where getting rid of androgen receptor would mean that their uh, proliferation will be badly affected. And rightly so, because we know that both of these genes, LNCAP and 22, both of these cell lines express very high levels of androgen receptor, and they are dependent on androgen receptor. That's why we call it dependency map or DEP map. Uh, as opposed to others, you can see there are very little effect whatsoever on other cell types. Does that make androgen receptor a good target? Yes. Why? Because if you're going to make therapies, if you're going to synthesize drugs that block androgen receptor, they're going to have a very bad impact on prostate epithelial cells, cancer cells, but they will not have very much side effects on other tumor types which do not express androgen receptor. So you can see getting rid of androgen receptor in these cell types, ampulla of Vater, biliary duct, bladder, bone, does not really have much impact. So you can get some first-hand evidence of the impact that the knockout perturbation of particular gene A, B, or C will have on your cancer type, or even normal cells. For example, eye cells, fibroblasts, head and neck, kidney. By the way, these are mostly the cancer examples like head and neck. You have some data, how many 77 data sets within this. So this is how you're going to do this analysis and identify uh, first hints. I'm not saying if this data looks negative for your protein, don't go for it. What I'm saying is it will give you a good idea to do some sanity checks and to reflect whether the gene that you want to study as part of your PhD in particular cancer type, is it likely to have any relevance there, or this is completely out of question. This is something you can get by studying it from using the DepMap platform. Any questions on this? So what we are developing at the moment, you can see here, this platform allows you to study only one gene in a different cancer types. Because my lab studies mostly prostate cancer, we have downloaded all the data which is submitted to DepMap portal. And thanks to my computational biology colleagues who've done this for us, we are able to study any number of genes in cancer and the effect they have. If we want to study 200, get some idea, there are 200 genes. There seems to be a signature that I have been asked to study. 200 genes, that means you sit all day long and you want to study on prostate, and that would mean it will take a lot of time and you will not get comparative data, meaning getting rid of androgen receptor kills these cells, and you can see the gene effect is shown in -1.4, expression 7.59. So very high expression, and gene effect is very, very high. These are log values. You can see very high expression, and getting rid of androgen receptor would kill those cells. But this is only for androgen receptor. If you were to do it for 2000 genes, for example, 200 genes, it will take a long time. So what we have done is developed a platform that allows us to do the same, but do that only for prostate, but for multiple genes. Uh, this is still under development, so I won't be able to show that to you. That's one thing. So that gives you first idea of your gene you want to study. I wish you would have told me a gene if it was not a secret. Then I could have shown it to you. One of you could have shown, but I understand you can do the same analysis yourself because this is being recorded. So Padmavati is asking, are those maps based on experimental studies? 100%. So these are all based on, I should have mentioned this, this is all based on CRISPR screen, knockout screen of CRISPR. And you can see here, this is the CRISPR knockout screen that is showing here that we are using. So this is the CRISPR map, publicly available data set that we are using, Padmavati, in this. So, for example, androgen receptor sounds very good. If
Dr.
you want to
Asif,
study in,
is there
yes,
any
please.
way that we can know about the off-target effects, if there are or there are not?
Off-target effects of the expression. I think what you would want to do. So, this is an excellent question, Dr. Aziz. With this, you get, because this is a screening data. It simply means it has not been validated in vitro. So I will take this data with a pinch of salt. This
Yeah.
is th- this is not something you can take at face value. You have to do multiple other testings to make sure that no, this is not something you would want to study. For example, androgen receptor is expressed in breast cancer cells as well, and you can see gene effect. It is showing minus, meaning in breast cancer cells. Also, getting rid of androgen receptor affects their growth. But also, androgen receptor is expressed in liver cells. So let's see where the liver is. Have a look at this liver. This is again showing very little but negative effect of androgen receptor. Getting rid of androgen receptor decreases their growth, it seems. So there is positive expression. These cells are expressing androgen receptor. We know liver does express androgen receptor, and one of the ways people have tried to stop hepatocellular carcinoma growth is by giving androgen receptor inhibitors. And to some extent, it has been successful as well. But the prominent role of androgen receptor is only so far in prostate cancer and molecular apocrine breast cancer. So that's an excellent question, Dr. Aziz. You have to see what sort of data you can get. And again, the question of side effects will depend on whereas this protein is being expressed. Are they being reliant on this protein as well in terms of their growth? I hope that makes sense.
Absolutely,
So
yes.
thank you. So I'm going to show you another platform now, which is very good to see. Okay, there is protein which is being expressed in this particular cancer type. Remember, the data you're seeing on your screen is taken from cell lines models. This is not data taken out from real tumors. So if you want to have a look at the expression of androgen receptor in real tumors, is there any way that you can get that data which is publicly available? Yes, you can. And there is a platform which is called Protein Atlas. If you can please, I will provide the link here. Human Protein Atlas. So if we go to this platform, can you please confirm that you can see this page uploaded on? Thank you,
Yes,
Swati. So Protein Atlas is an excellent, thank you, Dr. Aziz. It is an excellent platform as well in terms of studying the primarily I use it, my lab uses it to study whether any new proteins that we get from screen mRNA, uh, are they even expressed at protein level? I told you that protein expression and mRNA expression are not linear, meaning if you see a lot of mRNA, it does not mean that you will have a lot of protein. So before you start doing that journey, and I understand there are inhibitions, antibodies are super costly in India. Um, qPCRs, getting primers is, of course, much more feasible and easier. It takes time from what I hear from colleagues in India. So you can actually do a lot of discovery research using this platform, preliminary research for your own project. And you can get actually, let me show you. So we use it to see whether proteins that we find in the screen, they're expressed, are they expressed in cancer. So for example, if I go to androgen receptor, You can see here in this platform, it has recognized that there is androgen receptor. You click on it, and it will give you a lot of data. Now they have combined it so that there are lots of other data from tissues. There are interaction data. Remember what Dr. Aziz was telling you, pathway analysis, and there are interactions between proteins. That is what it is talking about. The three-dimensional structure of the protein, and so on. I want you to have a look what happens in the real tumors, whether they do express androgen receptor or not. You can study and treat cells in cell culture. You can kill them, but is your protein in fact being expressed in cancer or not to have any clinical relevance in the first place? This is something you can do by using this platform. So if we go and click on cancer, uh, it looks like this is showing you the TCGA, the same data set I showed you previously in Xena browser. I'm going to show you the Xena browser in a minute. Have a look here. It looks like androgen receptor is highly expressed in some forms of brain cancer. It is expressed in some forms of prostate cancer. Also in breast cancer, androgen receptor expression, transcript expression at least, is very high. So if I go on prostate and show you some slides, in fact, that is what you can access from this platform. So if I go to prostate, you can see immunohistological, immunohistochemistry data from these slides. Have a look here. They have patient data, core by core, provided for patients stained with androgen receptor, and see the expression level yourself. So you have to be a pathologist to understand these slides. I am not one of them, but I can tell you based on my experience and what I'm seeing here, these are the glands that are the ones that give rise to cancer. These are the glands within the prostate tissue. What you're seeing here is a stroma or connective tissue. That's what we discussed before, where the connective tissue is. I was showing you a cartoon, and these are the glands, some of which are going to make cancer. You can see the cells expressing very high levels of androgen receptor, shown in this brown stain. Uh, it says very high staining, and you can see if you were to do staining to do the nuclear stain, you would see that the staining will nuclear staining such as DAPI will tell you that these oval blocks are actually nucleus. What you're seeing here, can you see my cursor?
Yes, sir.
Thank you, Swati. So what you're seeing here is actually extracellular matrix, but also, the lumens, the ducts, that's where the secretions are going from these cells. But also nearby, what you're seeing is a cytosol, which is not very visible in this picture. So what you see is that androgen receptor expression is largely restricted to the nucleus. That's what we are being told by this platform. And you can see here, you need to be a pathologist to identify whether this is cancer or not. In this core, you can see there are some cores which look like they have lost the structure. So normally you will have a two-cell layer. One of them is the luminal cells facing the luminal. The other one sitting on top will be the basal cells, and that is the architecture which is abrupted, which is gone in prostate cancer. So I would say this is prostate cancer. And moreover, what I can say for confidence that indeed, yes, this protein that I want to study and spend next 3-4 years of my life doing a PhD does indeed express androgen receptor. This is androgen receptor, which is very well known. You might apply this knowledge to study a new protein you want to study in order to have its clinical relevance identified, deconvoluted during your PhD. And that means you would know that what you're doing may one day actually have a clinical impact, because the protein that you're studying and trying to block using understanding the pathway that actually activate this protein may actually have one day effect on the clinical treatment of the disease. So that is something I extensively allow and encourage people to use to have a look at the expression patterns and of your protein of interest. What it also does, it tells you some antibodies that will definitely work in IHC protocols. So they do sell some antibodies. They do recommend some antibodies, not directly the platform, but they will tell you mostly from Sigma where you can get some antibodies which will work very well in identifying cells which express your protein of interest, in this case, which is androgen receptor. So I hope that is clear. I can go back. You can see there are different codes from different patients. Of course, there are no patient names, but what you can see here is that they have been coded. For example, have a look at this. All you see is mostly stroma, but you see there are some normal and cancer cells, cancer epithelial cells as well in this slide. So that is something very good that I would recommend, highly recommend you use. And the last platform for today that I'll talk about is likely to be Xena browser. And let's see what we get from information. So what I would try to do is to replicate
I've
what I
already
will
posted
discuss. Sorry,
I've already posted the link there,
Dr.
but
Aziz,
it's
you were
showing insecure.
for Xena, yeah. Okay, let me do that. See if you can access this through this platform, xenabrowser.net. So this is the ultimate platform we have to do some discovery research on clinical cancer genomics. It may sound like slightly complicated, but I'll try. I've tried before with my omics tutorial to make sense of it and make it easier for you to understand. So first of all, this is a platform where patients, the samples, especially the transcriptomes, have been submitted, and the expression has been arranged into high expressors versus low expressors, and you can do it at a single click based on any protein or group of proteins you want to study, and you want to see what is their correlation with each other. So I was giving you an example where I was showing you the androgen receptor expression of ARID1A, the protein that binds to androgen receptor, and I was showing you that if you have low androgen receptor, you have low ARID1A. But there was another gene, A1BG, which seems like follows the opposite pattern. So first, what we will try is to replicate those findings using this platform. So let's carry on. So first of all, that study was prostate cancer. So if you go here and select this study, I hope you all can see that. What you can do is select that study. That was PRAD. PRAD stands for prostate adenocarcinoma, or short for prostate cancer. So you can select this study here and use it. This is the study. You can tell the system, I want to study. I want to see what is happening with the gene that I want to study during my PhD, for example. So you can see here, there are still 568 samples, the number of samples in this data set. And what you want to study is the level of mRNA for androgen receptor. And you can tell the system which database do you want to study. And to start with, you can simply say, I just want to see gene expression, meaning what happens to mRNA of androgen receptor in patients. If you do that, this is going to, again, put these patients into high expressors. You can see these patients have very high level of androgen receptor transcripts. These patients on the other side of the spectrum have very low level of androgen receptor. And now you've been given a PhD project where you want to see your gene of interest expression with, let's say, androgen receptor, and compare it with androgen receptor. All you have to do is type in the name of your gene. So A1BG, as you can see, stands out here. That's why I used it as one of the genes. But I'm going to show you the gene which is known to be a partner for androgen receptor and regulates androgen receptor activity. And that will show you that actually these are part of the same pathway. ARID1A gene, and we are again going to do basic analysis, gene expression, and click done. Have a look here. This is one of the finest overlap you will get. So you can also zoom in, and if you want to zoom in, just hold your browser and click here, and it is going to zoom in your data. So these are now, you can see the scale has shifted. Previously it was 50 samples. Now it is telling you these are 5 samples. So you can see very neatly patients who have very high level of androgen receptor across this board likely have very high levels of ARID1A. Why this is important? Because we understand we cannot target androgen receptor in prostate cancer. It becomes mutated, it becomes amplified. Fair enough. We know that genes change, gets mutated in cancer. But perhaps we can target what makes androgen receptor active. In other words, ARID1A. And in fact, people have done this. So they try and target ARID1A using drugs that will block ARID1A function. It is noteworthy. It is not important only in prostate, but it is also important in driving some forms of breast cancer through estrogen receptor, because androgen receptor, estrogen receptor, they're very analogous in terms of their activity. They're both nuclear receptors, and it is known that ARID1A can support prostate breast cancer growth as well by binding to estrogen receptor. So you can ask this question: is this the only gene which is highly expressed uh, or co-expressed with androgen receptor? There is a separate platform. If there will be interest, I can show you that platform today to see, okay, my gene is in cancer is gene A. I want to see this. We already know that ARID1A is overexpressed in cancer because it is a partner of androgen receptor. How about that gene which shows the maximum activation or maximum follows the same pattern of expression as androgen receptor? ARID1A is one of them, but is it the top 5 genes, top 10 genes? There is a separate platform that will allow you to do that. So this is what we were showing. I showed you in the omics tutorial. You can zoom out again or clear the zoom. So if I clear zoom and type in the name A1BG, let's see what you get. That was the name I showed you. This is a good prognostic marker for liver cancer, and study the expression pattern of A1BG. You should be able to essentially replicate what we were getting before. Yes, that patients who have very low levels of androgen receptor have very high levels. I can zoom in and show it to you. Have very high levels of A1BG. Have a look here. Very low levels of androgen receptor, very low levels of ARID1A, but very high levels. Most of the patients have high levels of A1BG. That should suddenly tell you something. You can start uh, deriving some conclusions based on this. Perhaps androgen receptor, as I was telling you, is repressing the expression of A1BG. And you might want to go back PubMed and check whether there are reports of A1BG being regulated by androgen receptor, where androgen receptor serves as a repressor for this A1BG gene, which we know in liver cancer is actually a very good gene. High expression protects against liver cancer. Perhaps this might be similar case in prostate cancer. Who knows? Because androgen receptor blocks it, and we know androgen receptor promotes prostate cancer. So if you connect all these dots, it may be that androgen receptor promotes prostate cancer by blocking A1BG gene. So you can actually get a lot of data, very good publication quality data, or at least to start with your PhD, a lot of data for your synopsis to think about what you want to study in during your PhD. So I'll clear the Zoom and stop this here, the Xena browser. You can see ARID1A is one of the genes which is highly expressed, shall I say co-expressed, and follows the same expression pattern as the androgen receptor. Dr. Aziz, will I have 10-15 minutes to take questions, but also to show them a platform which will allow them to find out which genes are the most expressed top 10 genes which are most expressed with their gene of interest. Uh, added 1A, I knew it, so I showed it to them. But that platform will tell them actually there are maybe part of same pathways and follow the same expression patterns. Okay, so let me take questions first. I'll take some questions first. Are there any questions? Please make sure your mic active and ask me any questions. Padmavati is asking, can we use data from these sites for publishing papers like meta-analysis? Yes, 100% you can do that. There are specific journals which will take such data and publish. You can publish it. Normally, for any good publications, may I suggest you need some elements of in vitro validation, some molecular biology validations through some experimentations to publish them, because right now there are parts of the world where this is becoming quite common. People have developed tools to bring all of these platforms on a single portal where they can access and uh, basically churn out a lot of data in no time. And as a result, there are concerns that what is the meaning of these data if it is not validated through experiments. And therefore, there are journals which will decline to publish anything which is 100% computational biology. Fair enough. But yes, there are journals who still and very well welcome data. They're low impact journals, but they do take data only as computational analysis for publication because they are publicly available. If you do your own analysis, like I showed you the paper yesterday, the mutation paper, the air pollution paper, nature paper, most of it was basically computational data from patients, but that was not publicly available. And all this those authors did was to analyze it. No, that was their own data, and they took time to do lots of extensive analysis, uh, showing some signatures coming out of that data and publish it. So yes, if you have your own computational biology data, that will certainly lead to very high impact papers if very relevant, and of course it depends on the journal as well. I hope that answers the question. Any other questions? I can show you then a platform. That will be the last platform. I'll come back again to showing you the Xena in a minute, but I want you to have a look at this platform called, I'll keep this like this, it's called GPR2 platform. So I will give you the link in the chat box. Okay. So in this, looks like it is not working for some reason. Okay, I don't think this is working, this platform. So you can study a lot of your proteins of interest using this platform. I was telling you that androgen receptor interacts with this ARID1A, and a lot of proteins, in fact, interact with a lot of other proteins. There are actually interaction maps. Dr. Aziz was showing you some examples. There is a platform where you can actually access publicly available data sets like the proteomics data sets coming from mass spectrometry. And one of the platform is called the protein uh, string data set. Have a look at this in your own time. This is an excellent data set to tell you the protein that you're going to study during your PhD, your MTech, your master's, what what are the binding partners of your protein of interest. This is an excellent platform, and I would suggest that you have a look at this platform. Uh, so you can see here, this is a database of all the protein-protein interactions. They may be physical interactions, it may be one protein regulating the expression of other, or vice versa. This is what you're seeing on your screen. And what is happening here is that this is the data for over 12,000 organisms. How many proteins interactions are there? Over 20 billion. And how many proteins? 59 million proteins. So you can see how diverse the protein numbers are in different organisms. They may be paralogs, they may be proteins which are alternatively spliced, or altogether they may be totally new proteins, who knows. But you can actually identify, when you start a project, you can identify proteins that interact with your protein of interest. All you have to do simply is click this button, search. And if you go to the protein name, you can type in the name. For example, if I type in a wrong name, such as androgen receptor, it will not identify anything, meaning there isn't anything like that. So you need to identify the correct name. Uh, oh, it's still not showing. So let's try that. And Homo sapiens, which we are, and type in search. Let's see if it finds something. Yes, it has found something. This is what we are talking about, androgen receptor. We are not talking about any other thing. So if you go and this is the one that you want to study, you can continue. If this is not the protein that you want to see, what is binding to my protein of interest, you might go and have a look at other proteins of your interest if they are the ones there. So if we go to the androgen receptor in Homo sapiens, we are not talking about any other species, and continue. Let's see what you find. I believe this is not something which you have been discussed. Uh, and have a look. These are the proteins that it is showing. Androgen receptor is known to bind. So you can play around with the binding. You can make this extended, see what are the core binding partners of androgen receptor, but take this with the pinch of salt, as I mentioned. This is the data which is what we have found. Androgen receptor is to interact with 1100 other proteins. This is only a few proteins that the data is showing, but this is a rather still quite important, because it tells you that these are the proteins that interact with your protein of interest. If you're someone who's doing proteomics studies, you can certainly use the this link. You can register and log in to upload your own data set to study them through the platform, to see whether there are any patterns of interactions. For example, here, these are different nodes that tell you what is happening. One of the predicted partners, like I was showing you with Xena, what was binding to the androgen receptor, ARID1A, was following the same patterns of expression as the androgen receptor. In this case, this data will tell you what is the predicted functional partner, meaning the protein that is likely binding to the androgen receptor. Have a look at the first one, HSP90, which is nothing else but a heat shock protein or a chaperone. What is the score the system is predicting for the binding with androgen receptor? It is saying 0.999%, 999. And that means this is the most score you can get, is 1.0. You don't see it, but normally this platforms give you the score if it is high binder as 0.999, which tells you this is very likely a protein that binds to the protein of interest, my protein of interest. And indeed, this is the case. Chaperones, HSP90 family chaperones, they bind to androgen receptor and stabilize it. It is very well known. And then you can actually study what is the evidence for this. And it says there are experiments done. So what it is doing, this platform is curating a database based on the real research done, and then identifying, okay, let's see what is binding. So these are experiments done. What is binding And this experiment, there must be experiments done that show androgen receptor binds to this protein. There are databases. What these databases may be, they may be mass spectrometry databases that suggest binding. And there are text mining as well. So evidence, people making meta-analysis. There are overwhelming evidence to suggest that androgen receptor bind to this protein of interest. So there are few things that you can do at the very early stage in your project to make sure in the end you will have a very robust, meaningful project that may be helpful in saving lives of cancer patients. So with this, I'm going to stop and take any questions if there are any. So I'll give 10-15 minutes now. I didn't give the break before. To those of you who want to stay behind and ask me questions. Swati, are there any questions?
So there is one by Padmavati Ganeshan.
Okay, I
Um,
can't see. Yeah. Okay, yeah, she's asking a question. Sorry, I missed that. Can we use the data from these sites for publishing papers? So I already gave the answer for that. Looks like there aren't any more questions. In which case, shall we close the session, Swati?
sure, sir. Sure. Thank you very
Okay,
much.
obviously, thank you for your time
So
in
we just
organizing
got
this.
another question.
There is another question. Hamida is asking, Walaikum Salam. I've recently completed Masters in Biochemistry. I have only research experience of dissertation. Do I need to solve more labs or can directly apply for PhD? Do you mean a PhD abroad, outside India, or do you mean in India? Uh, uh, So let me answer Hamida's question. First of all, I'll come back to you. Basically, if you want to do PhD in a good place abroad, as an Indian national, your best bet is if you want to save time and not spend multiple years doing coursework and go to America, which is of course not very permissive these days, as you might have seen. I would say Europe is a very good place to do a PhD, and lots of students go there. If you have, so for example, when I went to do my PhD 22 years ago, they did ask me. The form asked me whether I had qualified GATE and NET because they do. Even back then, they recognize those qualifications as being very competitive. So there were, of course, a preference given to students who have qualified these exams. So that was one element. I had written a project on androgen receptor 1 year before, of course, so I knew about androgen receptor a lot. I did not have very much lab exposure on working on androgen receptor, so I mitigated that not having the research experience by writing paper, by writing the research grant application uh, and submitted it with my future supervisor. That's what I did, and that was successful. But if you're asking me with regards to you completed a master's in biochemistry, have research experience in dissertation, see if it is possible for you to publish a paper from your master's work. I'm not talking about a first author paper, but I'm talking about paper as a collaborative research with other people from your master's lab, perhaps a research article which you may have published. This might be little bit helpful. Certainly no issues with that. But if you want to do a PhD in India, certainly having JRF would help. I know there are still lots of competition with JRF, that even after JRF, students may not get top labs that they are after. For that, what I would suggest people should do is to join as a research assistant in a top lab, a lot labs that publish very good papers and where they will not be cleaning the glassware, but they will be doing some research for a year or so. And that means if they've done this, they have substantial research experience. They can expect, they can apply extensively. For example, countries like Germany, Belgium, France, to do a PhD there. Uh, if you publish any papers or have substantial experience, I get lots of CVs from many countries, including India, and the first thing you see is, okay, you've done CRISPR, you've done Western. I believe you've done Western. I don't think you're an expert of CRISPR. Oh, we have done RNA sequencing. Well, I don't really, because there is no evidence, there is no publication which will substantiate what this student is telling me. I take that with a pinch of salt, and there are plenty of people, sadly, who can write references without full honesty, and that's why you have to be very careful, because it is a big commitment taking a student on board. So if you attach yourself, there are institutions in India, NCBS, IITs, and of course AMU, IIT, IISERs, where there are people doing excellent research. Become part of them. If there is any position for a year, engage with the research, publish some papers, and that will mean, by investing that time, even if they're on a voluntary basis, if you invest 1 year, after that you work very super hard, chances are that you will be able to then apply and be expected to get shortlisted at least for the interview. Then it is up to you if you're able to answer questions in an interview, if you talk, and if you don't talk. That's why I'm asking you to ask questions to me while you're here, because that helps certainly prepare you for an interview. So I have another question from Mustafa. What is the future of AI in this field? Well, AI has limitations. Remember, it works on machine learning. What you feed AI, it brings you that out. It does not have a brain of its own. So no way this can come anywhere close to human intelligence. It can certainly facilitate research. For example, the platforms I was showing you, they're all, many of them are built on machine learning. So certainly that helps, but there is no equivalent to human brain and what it can achieve. Certainly with AI, it might become, relatively faster to do research, but when it comes to doing groundbreaking research, which will change the course of treatment, for example, for cancer, it is going to be largely dependent on genuine thinking, intelligence, and hard work. Uh, Outside, yeah, Hamida, I hope I've answered your question. I'm also working on a dissertation paper. Yes, Hamida, make sure that you publish a paper in a journal which is indexed on PubMed. As a first author, that will be important. That will at least hopefully get you shortlisted. I can give you some idea. I think I applied for 1,700 places when I was a student, and got shortlisted for few. But one person did offer. There are lots of students applying, especially from East Asia, to come to European Union countries, and that's why there is a huge competition. Sometimes, I mean, even for my lab, you see students who, as a BSc students, have published papers, which is, of course, you can see how high the bar is being set. I'm not saying don't apply to UK, do apply to UK as well, but mostly to positions, for example, in labs which are Wellcome Trust funded or funded by Cancer Research UK, because they provide fee for international students. Sadly, most of the PhD studentships in this country do not provide tuition fee for international student, which is normally 3 to 5 times higher than the fee for British nationals, home students. So, are there any more questions, or shall we close this session? I don't see any more questions, Swati.
Okay, sir. Thank you, sir. Thank you very much for the
Thank
excellent and
you.
insightful lecture and the tutorial. So
Thank you very
with
much.
yes, sir.
Yeah, go on.
Okay. So with this, we conclude today's session. Thank you to all the speakers and participants for your valuable contributions and active participation. And we look forward to seeing you tomorrow.
Thank you, everyone. Take care. Have a
Thank
lovely
you.
evening. Bye.
you.